Welcome everyone. In this topic, photometric. We are going to study photometric. So, photometric very important biological system. The slide is photometric introduction, colorimeter, Lambert Beer law, part of photocolorimeter. This topic details. This slide show introduction about photometry. The photometry is science of measurement light is specific focused into electromagnetic radiation, visible light, human visual system, lumen flows, and canadial intensity. The measurements are visibly light, pressure of brightness by the human eye. The light and concentration within the limited human vision, concentrating by factor in brightness, colors, and precipitation intensity. The manometer 380 and 740 manometer, the photometer wavelength roughly. The measurement light of previous day, human eye, focus in luminance, intensity, flash and luminance. The measure of total electromagnetic radiation, photometer weight measurement according to the CIE, function of matching human visual insufficient. Now we move to purpose of branch of optical measurement, brightness of light taken into the account is that human eye is not equally sensitive to all wavelengths of visible light. The weighted of luminance function application biochemistry measurement interest in light PM transmitted through the color light solution and light in addition in the measurement luminescence. Photometric radiometric, which unlike radiometric which measurement absorbs energy, at wavelength photometric only study in the visible spectrum. Now we study the photometric equivalent preferential measurement of proper light, the precious human eye. There is several photometric quantic and this is listed of table below. The light focusing luminance M2 or LUS, style focusing luminance intensity canadian, luminance canadian M2. The surface luminance large length of focus. But luminance flow is a total luminance powers emitted source of all direction unit of luminance M. Luminance intensity, the light emitted from source is given direction, formula candle power unit of candle ECD. Luminance measure light of falling into the surface. Luminance property surface emitted unit is CDM2. Photometric quantity and definition. Photometric is a unit of symbol. The photometric quantity luminous energy, quantity visual impression light measurement is the energy. Luminous SI unit, luminous second or tablet. The tablet luminous flash, luminous intensity, luminance, luminance, luminance exposure, luminance effective. The quantity the visual impression of light measurement is energy. Total amount of visible light emitted all directions luminous energy per time lumen. The measurement how to the light of source produces visible light, definite touch, and luminous flash power consumed by the light of source, luminous per watt. The luminous exposure, luminous energy incident of surface per unit area, luminous second, LS. Luminous per unit solid angle per unit project source, candle per square meter, CDM2 or NIT. The colorimetry is working principal photometry. A colorimeter is used to test concentration of solution by measurement is observing specific wavelength light. The colorimeter determines that the specific reagent used to react to the specific compound from the colored complex. Concentration of compound is the pigment. The specific spectrum of light takes rest of light transmitted from pigment. The light detected to colorimeter detector. The optical density calculator OD is equal to 2 minus logarithm percent DOD direct proportional concentration of subject. The function of colorimeter. The function of colorimeter, color combination wavelength varying tends of produce of some light of frequency. So equal present all wavelengths across a visible light spectrum. The function of the colorimeter determines what quality of color emitted from solution. The polychromatic light, monochromatic light. The light source of light lines natural filter, cubic photo cells and output. The result is the wavelength. The law of illumination under the specific condition may the high concentration luminance dependent breakdown. The certain particular matter of catch-up division, the light used must be the single wavelength. The application determines concentration colored metal ions, biological simple like protein or glucose concentration. The monitor chemical reaction react. The principle of colorimeter is the light focus in monochromator cuvity, but adjusted perimeter light focus sample proto rejector sample which a proton rejector to amplify to output the several sample solution the concentration of first preparative evaluation concentration now unknown sample. The plotting the various observation graphic yield the calibration curve. To determine concentration result now sample concentration to sample and curve. Application by colorimeter. The embroidery print in the industry evaluation the cultivar printing the ink and paper. The food processing industry. The laboratory and hospital determine biochemical composition of sample like blood, urine, cerebral spinal fluid, plasma, serum. The textile and paint and industry. A diamond dealers is also a physical characters of bracelet stone using the colorimeter. Instrument also employs cosmetology in measuring ultraviolet protection level of skin care protection. The use to the world to the water purification, clean, present chemical like iron, fluorine, chloride, molybdenum, etc. The employed evaluator color controlled brightness, screen to mobile TV, computer, and television greater than visual experience. The colorimetry also employed in the pharmaceutical sector of part in good medicine. Blood sample tested using colorimeter to determine the amount of hemoglobin present. Now we move photocolorimetry instrument used to measurement observation light in solution determine the concentration. The lens monochromator, sample holder, detector, photo cell photo and display the unit reading optical density of transmitter. The colorimeter, part of the colorimeter section, sample fluid, wavelength reaction, observation reload, mode indicator, the system of colorimeter experimental wavelength control the degrees increase the system. The colorimeter operation projector. So system power system knob turn clock direction towards the light. 50 minutes time of colorimeter occurs to stabilize the light source at the detector. The wavelength control knobs are provided wavelength. The transmitter and the switch display mode is the transmission. The T factor to 0.0% make adjustment to ensure the sample chamber is empty, the cover securely closed. Until they reach the top of the trickle and the side of screw. To treat the fluid, the fingerprinting on the screw with extrude web and the web. Able to travel to result on a corona reading, place the gentle but completely onto the screw camper with the vertical guide. The line factoring in the direction to right. The guideline on the screw lean the camper rotating the screw 90 degrees of clock rotating. The cat is the code, close the compartment cover. 100% using transmitter observation control of knob. The status indicator to read the observation display the should indicate 0.0 if the transmitter calibration up down correctly. The transmitter observation control knob that does not do already the now value. The display back transmitter is the mode of key. The reversible previous pressure remove the screw. Compartment load 90-degree controller clock before the doing. The directly digital display the read present D value. The selector option using the mode key. Then the value directly from the digital display selected transmitter of mode. The screw to remove from the cable compartment. Close the compartment cover. Light source. Light source light produces energy and intensity to cover up the enter the visible spectrum 380 to 780 nm. Measurement the visible spectrum nearly infrared range. The suitability of measurement is ultraviolet range 200 to 900 nm. Slide. To reduce unwanted spray, the light flow in the light beam to the pass through. Condensation lens. The parallel beam and light emerging from condense lens after the light passes through inside the integrated node. Monochromated. The monochrome light, polychromatic light, observe unwanted light wavelength and emits one monochromatic light. These are the type of prism, gradient and class. Prism refraction for light, it is a pathway the medium another. Application of colorimeter. The colorimeter utilizes laboratory hospital expansion biochemical sample during cerebrospinal fluid, plasma, serum and more. The production of paint. Industrial also make a colorimeter. Employed quantity analysis protein glucose biochemical compound. Testing for quality water. Measurement concentration of hemoglobin in the blood. Basic principle Beer-Lamb law. Photometric measurement is generally passed to observation light colored substances as solution. The transmitted light indicates the exponential concentration thickness but the length of the colored substance increase. Directly proportional concentration of high light percentage of sample. Beer-Lamb light absorption directly proportional to length to the concentration of solution. The observation is molar absorptivity, bath length and concentration. The light focusing by high density of power of length. The beer lamp also be later to attract attenuation light of proportional material through light of traveling. The quantity analysis ultraviolet with photoscopic statement. The solution directly proportional concentration of observed speculum but the length to bm to through the solution. The mathematical A observed good analyze differential A logic logarithm 10. The molar of the molar intensity. C concentration of solute. I bath of length is sample. The relationship of transmitter diffraction into light of passes through. Lamination of formula. The concentration of sunlight is strictly usually 0.01 m. High concentration electromagnetic interaction between the molecular cause of division. Chemical division, change of analytic oxidation, dissociation, solvent is after the observation spectrum. Too much light of source of polychromic division occurs. The interest solution high concentration can obstruction molar observability. Important of beer lab. Additional usefulness chemistry beer lab problem to physics, medicine, microbiology. Remember, apply the from electromagnetic reaction to visible light. The beer lab into solution concentration help association polymeric reaction. The medicine when taken to apply the measurement amount of bilirubin and blood sample. Where the chemical food track finding the concentration application. The meteorology be alarmed at solar radiation in the Earth's atmosphere. Part of a photocalorimeter light source digital LED lamp prevent lens apparatus. The constant lens created in parallel and concentration beam. Monochromator filter. The light passes through solution filtering unwanted light. The past glass plastic hold the solution the analysis. Photo detector, photo cells. The light transmitted through the sample and converted into the electrical signal. Galvanometer and display a signal to the detector to display the result. Observation percentage of transmission. Now we move the made of the graph into which separated light of different wavelengths. A sample cell from the filter passes through colored sample solution plastic fluid. The range of frequency rectangular round and have a fixed diameter like 1 centimeter. The type of the substance is made glass, quartz, and plastic weight. 340 nm length. Flexibility enters about the length, ultraviolet and visible range. Each secreted and shorter lifespan. Photo cells. The photosensitivity to measure light intensity of converting light energy into the electrical energy. Galvanometer the signal photocells detect the measurement to galvanometer display the optical density OT and percentage of transmission.